Good morning. Very few of you were up, and sorry, I got so close to the camera there. We're up and ready to go this morning, and so instead of taking up the time for those 3 people's day, what I have done is sent them on their way, and I'm just going to record this lecture and post it for you in Canvas under Carbon, Unit 4, Carbon. You're supposed to be doing, well, let me just show you. You are supposed to be, I'm supposed to be in the right place, and you have your carbon chemistry quiz due on, oh, what do I know, Saturday in your lab modules under Blackboard. It's the lab 3 under exam 1 materials, and then you have these assignments, chapter 3 reading and homework, the special properties of water video, your water lab that you can do at home, that you are doing at home, and your water quiz. All of these are due by Saturday. Don't wait until the last moment to do all your work. I am busily this morning preparing for your next week. It's going to be our last week of content before your first exam. And so your first exam will cover chapters 1 through 5, yes, in your textbook. And we will cover the chemistry, the basic chemistry of life. So atoms, water, carbon as the functional backbone of life, and then the 5 for major macromolecules. So that's what you have to look forward to. Biological macromolecules next week, which is lipids, carbs, nucleic acids, and proteins. And then exam 5. Today we're talking about carbon. Nope. Where's my face? Sorry, let me share my screen. Today we are talking about carbon. Carbon itself is fairly easy. Sorry, I have both my monitors on and it doesn't play nicely when I do that. So let's do it this way. Still not what I want. That's fine. Carbon itself is fairly easy. Carbon is the building block of life. We have to have life and water. Water is essential to life, but water is not living. The water that you drink is not a living entity. Water is essential. Water doesn't create life. Life is created through building things off of carbon backbones. That's what a majority of life as we know it is created as, and it's life as we know it. We are carbon based on this planet. Why do we study carbon? You know, I'm so sorry. Just one more time. Let me just do that. There we go. Why do we study carbon? Because all of life is built on carbon. I just said that. All of life as we know it. Remember, this is a biological science and we cannot guarantee everything 100% because we do not know everything. So all of life as we know it is based on carbon. This is the backbone for our cells, which this is an animal cell up here. An animal cell with its nucleus and all of its organelles and a cell membrane. This is a plant cell. It still has a cell membrane and all of its organelles, but it is surrounded by a cell wall. That is the major difference there, plus the chloroplasts. So we are, 70-ish% water, but we are then 25-ish% compounds, carbon compounds, which are going to be your carbs, your lipids, your proteins, and your nucleic acids. That's how they're built into our body. Remember, you're mostly water. So if we took out all the water from you, you'd be very desiccated, like a mummy that's been preserved for hundreds and hundreds of years. Then if we pulverized what was left of you without water, you'd be mostly proteins. Then carbs and lipids, depending on what ratio you have them in, and then nucleic acids. And then you're approximately 3% essential vitamins and minerals, like sodium and chlorine, potassium and magnesium and iron and chloride and whatever. Happily, in your future, you have organic chemistry to look forward to if you are going to follow in the sciences. I found organic chemistry difficult, but now I know with much more maturity that I was not studying properly, but also I wasn't giving it the appropriate amount of time that it needed. I thought I would just get it and go on, and then I didn't, and I struggled in organic chemistry. but it's actually not a difficult science. Organic chemistry is the study of carbon compounds and how they interact with each other. Carbon is the building block of everything we have going on. They're very versatile building blocks. They're like your Lego sets that everything binds to and sticks off of, but they are the base Lego part. They will bind with nearly anything because remember, they have 4 valence electrons, and so they need 4 valence electrons. They don't have time to play the game of taking electrons or giving up electrons or trying to gain electrons. It doesn't work for carbon because it already has too many electrons, so it's not easy to give them up. And it needs too many electrons, so it's not easy to take them. So carbon does this very wonderful thing, excuse me 1 minute, where it shares. It just shares and shares and shares. It doesn't actually really care who it's bonding to. It just needs 4 bonds. So it'll make 4 stable covalent bonds. A lot of what we see in life is it just bonds to hydrogen very easily and happily. And then all 4 hydrogens will have 2 electrons in their valence shell, and then carbon will have 8, and everybody's happy. But it can bond, to many other things. That's fine. Here are different ways that we represent carbon and its bonds. You can do it in the molecular formula. CH4 is methane. Here it is in a structural formula. Each line represents a pair of shared electrons. This is a ball and stick model, which you can build. We have those building sets here in the classroom, and all the elements are different colors and sizes. So here the color of carbon is black, and the elements of the hydrogen is white. And then you have a space filling model, which is probably more accurate in terms of how much space the atom would be taking up in the molecule. Here's C2H6, ethane, same thing. And then here is C2H4. It has a double bond. Carbon can bond to itself. As long as it has, 4 electrons to share, it's happy. It can even triple bond. That's more rare. Look at this ball and stick model. Look at how this double bond is actually bent away from itself. That is because electrons don't like to share space. And so you can see here in the arrangement of these ball and stick and space filling models that the hydrogens are actually kind of being forced away from each other. This is due to s and p orbitals in those valence electrons. And so they actually push each other away. So when you have double and triple bonds like this, they're not nice, neat, straight bonds. The electrons are pushing each other away. The orbitals are taking up their own space. And so that's a more accurate representation of how the electrons interact with each other than the structural formula. Hydrocarbons are a huge class of carbon molecules in the human body and in biology. These are non-polar. I've had some questions since the last lecture. Does non-polar mean how polar it is, or does it have anything to do with polarity? No, actually. Non-polar and polar basically are talking about if it will interact with water. And non-polar molecules do not interact with water. The reason being that they don't have any charges for those partial positive and negative charges in water to interact with. And so they all just kind of separate from each other. Our hydrocarbons are our fats and lipids and anything else that is fatty or lipidy, like wax. And I'm not thinking very clearly right now. So they are hydrophobic. They are stable, and there's very little attraction between the molecules. And methane itself, this is a gas at room temperature. Oils are liquid at room temperature. Fats are solid at room temperature. That's actually the main way you tell the difference between oil and fats. And so you have that little bit of information before we talk about biological macromolecules. Carbons can grow. The happy thing about carbon is how it grows on itself. It forms giant chains. And you get these long chains of carbons, and they can be hydrocarbons, and they can be proteins, and they can be carbohydrates. nucleic acid, not so much. They kind of have a very set structure. But these long chains allow different things to occur. And so we can do different things with the properties of life with these carbon chains. And so you have ethane, it's 2, propane, 3. Then they can, so that's just a line. These are lines of chains. Then they can branch like this. And so this is butane, it's C4H10, but this is also C4H10 with a different structure. This is isobutane. These are called isomers. You can have double bonds, and they can be placed anywhere between the carbons. So it can be here where it is, it can be here in the middle. You can even have one over here on the 3rd carbon. And so 1 butane, this is number 1 carbon, double bonded. 2 butane, number 2 carbon is double bonded, etc. And then they can form rings, which is how most of our hormones are in our human body, in a lot of actual other structures too. They form these ring shapes. And so these are the different ways that hydrocarbons can grow. I talked about isomers right then with the butane and isobutane here. It's the same structural formula, so C4H10 for this, in this case, but it's different structures. So here we have C6H14. Here you have C6H14. Hey, here you have C6H14. They're all the same structural formula, but they have different shapes, which gives them different chemical properties and different biological functions. This actually really comes into play with medicine. And so you can get left and right, or L or R, structures of different types of medicine. And one of them can be functional in the human body, and one of them can be non-functional and or even harmful to the human body. So these structural differences create important structural or functional significance. For example, the amino acid alanine, the L-alanine is used in proteins. And that would be this one, sorry. But the R-alanine, oh, because it's Latin, so it's D, sorry. But the D-alanine, not R, it's D, can't be used in proteins. It's not functional. The structure, it's completely the same formula, which in this case would be, you know, C3H6N1O2. The formula is the same, but since the structure is different, it can't be fit into a protein and it won't function. In medicines, our L versions are active. So you'll have L-dopamine, something we take normally. Acetaminophen is an L. There's a lot of them. I'm going to be honest with you. The L version is active, but not the D version. And this is because of the way it interacts with the human body and can be broken down by our enzymes. Sometimes this has tragic results because sometimes that D version actually ends up being very harmful. Most of the time it's inert and doesn't do anything to the human body, but just doesn't function. But sometimes it causes incredible damage. And so, way back even before my time, we'll talk about that word stereoisomers and a difference. So I'm not as old as you think I am, but even back before my time, there was a drug called thalidomide. Thalidomide was given to women to prevent early labor. I actually think it was to prevent nausea, because if you have ever known pregnant women, they typically suffer from very bad nausea at least the first 3 to 4 months of their pregnancy. It's very normal, but it can be, you know, debilitating. It can actually stop you from living your normal day-to-day life. And if you know Princess Kate in England, she's had 3 babies, right? And every single time she's been pregnant, she's been hospitalized because she has extreme forms of this nausea, which is called hyperemesis gravidum. So hyper throwing up because you're pregnant is basically what that turns into. Anyway, so nausea is fairly debilitating for pregnant women. Thalidomide stopped that. And in Europe, it was approved. And for some reason, the guy in charge of the FDA here in America, he would not approve it. He would not approve the drug. Turns out that was a blessing. Because what thalidomide did, the D-isomer of it, caused severe birth defects. And so you got these very stunted children who had what we called flipper arms and or legs. These are the ones that survived. A lot of them just died either in utero or right after birth. But they had these severe genetic mutations due to the stereoisomer function of thalidomide. And of course, nobody knew that until like 9 months after it was prescribed. And then for about a decade, you just kept having children being born with that as they try to figure out why. It turns out it was the thalidomide. So a general surgeon, not the guy in charge of FDA, the general surgeon here in America actually stopped that from happening to American children. He's the only one who denied that being approved, but in the end, it actually saved a whole bunch of lives. Oh, let's just go back. So these L and D versions are called stereoisomers. They're mirrored images of each other. One is functional, one is not. Which just shows you, while carbon-based elements and molecules and whatever create life, they can also harm us if we don't use them correctly. The wonderful thing about carbon is it once again, it'll bind to anything. So it doesn't have to be hydrogens. It doesn't have to be oxygen or nitrogen. It'll substitute other atoms or groups around the carbon. You have something called ethane versus ethanol. Ethanol has a hydroxyl group on it. So it changes. It just takes off 2 of the hydrogens and puts an OH on there instead. That changes the way it functions in the human body. It goes from being a non-polar, I'm so sorry, I don't know why my mind is not working. It goes from being a non-polar molecule, because remember, hydrocarbons like this do not like interacting with water, to becoming a polar molecule, because this oxygen attracts the water molecules around it, and so it can become dissolved in liquid. It goes from being a gas at room temperature to a liquid at room temperature. Ethane is a gas used and produced for many various functions. Ethanol, it's alcohol. These are the differences. These are biological effects that occur just by changing the things that you bind to carbon. So we call these functional groups. And if you look at page 63 in your textbook, it has the major functional groups of biological life in it. And it's 1, 2, 3, 4, 5, 6, 7 of them. 2 of them are actually lumped together. They're ketones and aldehydes, but there's 7 of them. And we're going to go through them because you need to know them. Functional groups are parts of organic molecules that are involved in chemical reactions. Remember, whatever you put on the ends of those carbons is going to change the way that they interact biologically. So these functional groups give organic molecules, organic meaning carbon, distinctive properties. You have your hydroxyl group and amino group. You have your carbonyl group and sulfhydryl group. Carboxyl, so carbonyl and carboxyl, and phosphate group. And then this affects the reactivity. 1, 2, 3, 4, 5, 6. Fine. There's only 6. Carbonyls are actually 2, and they have them separated in the chart in 63. By adding on any one of these groups, you basically make hydrocarbons hydrophilic, and it means it increases their solubility in water. Sorry, I clicked too fast on that. I always think there's one more line than there is. Vive la différence, males and females. We're males and females based on a very specific set of hormones that are released or not released at the 6th week of development in utero. Every embryo, because it's not called a fetus until week 10. So every embryo in the human body starts out as a female. Then, depending on if they got an X chromosome or a Y chromosome from the sperm, differences occur at the 6-week mark. The difference being the production of testosterone. Testosterone creates male babies. Estrogen is females. They're basically the same hormone, right? They're carbon skeletons that have different functional groups attached to them. So you have the same structure here, a pentagon or pentacarbon with 6 chained rings attached, 3 6-chained rings attached to it. And look here, you have CH3 and OH, so you have a methyl group or methane group and a hydroxyl group. And then here's estrogen. It has a hydroxyl group on the end. Here's testosterone. It has another methyl group and then a carbonyl. And that's it. That's the only difference between male and females, is just the functional groups attached. And they cause all the major differences in physiology that occur between male and females, just by changing 2 functional groups. 2 tiny functional groups. Oh. So they interact with different targets of the body and cause different effects. That's why functional groups are so important. So you've probably heard about these functional groups before and forgot about them, and then heard about them and forgotten about them again, because that's how I did for a majority of my life. But these functional groups are actually essential to how our life works. So you have a hydroxyl group. It's an OH group. These are called alcohols if this OH is attached to anything. The name typically ends in "-ol", like ethanol, alcohol, methanol. Those are the common ones. The OH group allows it to become, liquid at room temperature, and allows it to become soluble in liquids because it makes it more polar. That's it for alcohols. You have carbonyls. This is the one that's a 2-for-1, because depending on where this C double bonded O falls in the carbon backbone, it's either an aldehyde or a ketone. So a carbonyl is just a C double bonded to an oxygen, a carbon double bonded to an oxygen. If you look at the chart down here, if this double bond occurs at the very end of a carbon chain, then it's an aldehyde. If this double bond occurs anywhere in the middle of a carbon chain, then it's a ketone. And that actually does cause very significant biological effects. I don't have any examples for you right now. Nope, and your book doesn't really expand upon it either. Carboxyl is both, it's a 2, a hydroxyl and a carbonyl. So carb from carbonyl, oxyl from hydroxyl, carboxyl. It's a C double bonded to an O, or a carbon double bonded to an oxygen, and a single bond with a hydroxyl group. You got C double bonded to an O with a hydroxyl group. These are known as acids. They're weak acids because they can lose that hydrogen or gain a hydrogen depending on if you need more hydrogen compounds in solution or you have too many hydrogens in solution. So they're a weak acid. Hey, look, it's vinegar, you guys. That's how we know it. That's one of the ways that you would know it, is this formula right here, which is vinegar. These are found in fatty acids and amino acids. Amino acids are the base of proteins. And one end of every single amino acid is going to have a carboxyl group. The other end has an amine group. What do you mean an amine group? See what I did there? See? It's a good transition. I'm very proud of myself for that. Amine group is an NH2. So ignore all the words in the middle. Come down to this example right here. This is an amino acid. So here's an amine group on one end. Here's a carboxyl group on the other end. It's an amino acid. It's a mean old acid. but this is how all amino acids are built. The only thing that's different is what's going to be coming off the bottom of the middle carbon. There's always a hydrogen, and the thing that comes off the bottom, we call it an R group because it's variable. Okay, so that's next week's lecture anyway, or lectures, you have 4 of them. So amino group is a nitrogen attached to 2 hydrogens. It causes amines. The NH2 acts as a base. So these can be weak bases as well in the human body because the ammonia, which is this, can pick up a hydrogen from solution and become NH3 plus, or it can release a hydrogen and stay at NH2. Sulfhydryls are sulfurs bonded to hydrogens. We call these thiols in the human body. It stabilizes the structure of proteins. You'll have something called it's sulfur bonding to a disulfide bridge. That's what it's called. And it's a sulfur that binds to a sulfur, and they're one of the strongest bonds in the human bodies. And so we have those in proteins and in DNA. And so they're very, very strong. But here's an example of ethane thiol, which I wouldn't even know what it does in the human body, but it has a sulfur and hydrogen sulfhydryl group attached to it. Phosphate, this is your energy. This is where we get all of our energy from, is phosphate in the human body, essentially speaking. Phosphate is a phosphorus bound to 4 oxygens. Connects to a carbon through an oxygen. So come down here to the example and you see that there is the phosphorus, then the oxygen, one of the 4, then the carbon. So it's a COP bridge. Lots of oxygen gives it lots of negative charges, and so it's super reactive. And it transfers energy between organic molecules with ATP, GTP, CTP, and TTP. Because adenosine, guanine, cytosine, and thiamine all have their own versions of this phosphate group. Was that 6 already? No, the last thing it doesn't talk about, but is a functional group, I mentioned it here, is these methyl groups. And methyl groups affects the expression of genes, I'm just reading out of your books, when on DNA or on proteins bound to DNA, affects the shape and function of male and female sex hormones. So, excuse me, this methanol group is also a functional group. Don't forget it. The last 5 slides deal with our topic next week, which is our macromolecules. Once again, our macromolecules are lipids, carbs, proteins, and nucleic acids. You actually have slideshows over all 4 of these, and it is my plan this weekend to record myself doing all 4 of those lectures. Macromolecules are big. Macro means big, and so smaller organic molecules join together to form larger molecules. For example, amino acids, small, form proteins. Proteins are giant. Nucleic acids form DNA and RNA. Carbs form carbs. Carbs form bigger carbs. Your 4 major classes, we know these are carbs, lipids, proteins, and nucleic acids. So you have to make polymers out of monomers is what happens. And you do that through something called a dehydration synthesis. You dehydrate, so you take water out. Long molecules are built by linking repeating building blocks in a chain by this dehydration synthesis, and then that forms a bond. These are covalent bonds that you form here, right? And every time you do this, when you create bonds like this, you're storing energy in that bond. It's potential energy, you know, potential versus kinetic from chemistry and physics. You put potential energy into the bond. So if you need to break that bond later, you actually gain energy. Sorry, this is just repeating what I've said. A dehydration synthesis takes out water. That's how you build it. How do you think you break it down? Oh, condensation reaction is another word for dehydration synthesis. It means the same thing. It does require energy and enzymes like everything else in the human body. Enzymes happen probably early October for us. There's a lot of rules regarding enzymes and how they function. We'll talk about how enzymes function, but you do have to have enzymes to take it out. There's that enzyme. You break it down by putting water back in. It's called hydrolysis. Hydro meaning water, lysis to break apart. That's how you break it down. So let me put all the words up here. There. It's a hydrolysis reaction or a digestion reaction. You use water to break down polymers. It's the reverse of dehydration. You cleave off one monomer at a time. And you just put that water, you put an OH on one end and an H on the other where it would have been originally. Also requires enzymes, of course. And as I said, again, every time you form a bond, you put energy in it. So every time you break a bond, you release energy from breaking that bond. So now I've just lost my mind. It's the last slide and I've lost my mind. Here is a man's impressive tattoo of a backbone and muscles, which have to be built out of carbs, lipids, proteins, and nucleic acids. Do you have any questions? Right, I'm recording myself, so you can't ask them. Oh, Lord, what a crazy day. Okay, if you have questions, I'm always available via email, 972-600-5320 before the end of the week or before 3:30 every day. So contact me if you have questions. Thank you so much. Good